Hello, my name is Sarah Ellis, and I'll be talking about Colleen T. Scow and the misconduct that she did with the data in her research that was retracted. So Colleen T. Scow was a PhD student from the University of Chicago. She is also a former postdoc fellow at the Cell Biology and Physiology Center at the National Heart, Lung, and Blood Institute, also known as NHLBI for short, and at the National Institute of Health, the NIH, which is where this specific research project was conducted that she did unethically. She also received a recruitment grant from the University of Texas Southwestern Medical Center for $2 million for a tenure track position. And that is what kind of encouraged her to pursue this research project and where she ultimately did her misconduct. So the research topic addressed foreign family actin filaments, also known as FMN2. They were discovered in the cytoskeleton to help prevent nuclear damage whenever cells go through tight spaces. It was found that FMN2 upregulates in melanomas, and so the different factors of the FMN2, such as the actin focal adhesion system and the FMN2, they help to limit DNA damage and envelope breaks. And they help to retain the shape of the nucleus and the cell position when the cell is migrating through tight spaces. So the results of the study showed that FMN2 plays a critical role in making the actin focal adhesion system and helps the cell survive tight movement. And so in cancer, this ultimately promotes metastasis of the cancer because the cell is able to survive. And so the study explored the role of the focal adhesion associated formins, and it found that FMN2 promotes metastasis of melanoma cells in lungs. So the research methods included using mice as animal models, specifically female mice, and so cell lines that were used were primary murine embryonic fibroblasts, human foreskin fibroblasts, human umbilical vein endothelial cells, B16, F10 melanoma cells, which were injected into the mice using a cloning system using a plasmid with cDNA for the murine FMN2 green fluorescent protein, and they tested to see if the FMN2 and melanoma metastasis in mice inside the living organism using tail vein injection model. And so after they injected the mice with this clone, or with this plasmid, they then euthanized the mice and then they extracted the lung tissue from them, and they stained the cells, or some of the cells, to do a histological analysis to see if the melanoma cells metastasize into the lungs. And then they also used the unstained lung tissue and mounted it on a slide to analyze the immunochemistry of it to see how the FMN2 interacted with the cell chemistry as well. So the misconduct that occurred was that Scow altered many of the results in the figures within the study. So first of all, she used fabricated primary data. So the data that was initially presented wasn't even a good start because it was all fabricated. It was all made up. And so she then selectively omitted and included data points in 10 of the figures. And she also falsely reported the statistical significance of a large amount of falsified data. So not only was the statistical significance incorrect, and she falsely created it, she also did statistical significance on made up data. None of it made any sense or applied to the study at hand. And this was across 14 figures in two different papers. And so an example of this was that she reported measuring 250 focal adhesion molecules pure condition, when in all actuality she only measured 56. So that's a huge difference in what actually occurred. And then she also fabricated and falsely labeled experimental results from alternate experimental conditions in 7 of the figures across 2 different papers. So she had, before this paper that we're talking about right now, she had another paper that was retracted because of the same reasons. And so she didn't learn her lesson, and she continued to do it in this paper where she was given $2 million as a grant. So the listed figures are examples where she omitted data or she fabricated and falsified data. And so this next slide shows 3 different figures where figure 1E is where she omitted data, and figures 2A and 2C is where she fabricated or falsified data. So sanctions that were administered because of her misconduct was 1, the research article was retracted, and 2, a voluntary settlement agreement issued on January 25th, 2018 said that she had to be supervised for the next 3 years in any other research that she participated in, and that an employer must submit in conjunction to an application for the PHS a certification from the ORI that confirmed that the research that she or the data that she gathers from her research is legitimate, and that she also has to voluntarily exclude herself from serving an advisory role for the PHS for the next 3 years. So the impact that any type of misconduct or falsification of data has on its authors is that the author is no longer trusted. Their reputation is tainted because of their actions and their misconduct, especially if their actions are repeated over the course of multiple times. So the co-authors that fall underneath them their reputations are also tainted as well, but not to as much of a degree. But it was shown in a study that co-authors of someone who has falsified data or fabricated data have fewer papers that are published after the fact, and also the number of citations of their work is decreased. Institutions are negatively impacted with this as well because it's a waste of research funds, and it discourages the PhD students that are in their program from doing more research with their peers because they are seeing a growing number in cases where people are misrepresenting data or making things up. And so in science, this is also detrimental because scientists and researchers lose trust in the scientific process. And they also, within science, continued research is sometimes based off of retracted papers. And so when it is found out that the research that a study is based off of is all false because of fabricated data, they have to redo their study, and then the project is also wasting their money and wasting their time, and it's hard to get all of that back because of the foundation on false data. So thank you for your time, and thank you for listening. Have a great day. Bye-bye.